So welcome back. And thank you for all the interesting questions in the last session. I really enjoyed it. But I enjoyed it a bit too much, I guess, because like I said, I had 3 bits and I thought in the first lecture I would finish 2 of those bits, but I ended up only finishing 1 of those bits. So I will have to kind of, figure out which of these I want to talk about. The second one is like the bulk of it is a case study with all sorts of complicated thinking of moving a model from like mouse to monkey to human. The third case study was not that complicated, but kind of high impact. So I feel like maybe I'll just give you a little bit of a flavor of what's going on in the landscape and, kind of skip over that complicated case study and then, talk about what I think is sort of very high impact work that we were excited to be a part of and kind of in the heart of clinical oncology. So I hope you all are able to take some of what we learned as you interpret clinical trials in cancer. So for the second bit, I'll just give you a little bit of an overview on what's happening in cancer these days. I think it's an extremely fertile area of research. Lots of exciting things are happening. And in particular, the 2-3 things that we are working a lot on are checkpoints, ADCs, and bispecific antibodies. My third case study is on checkpoints, so I'll get to that in a second. But I just want to talk to you about kind of almost the futuristic, like, very fancy things you're able to do with antibodies these days and what the modality of the ADC is and the bispecific is. So I'll just talk to you about both of those. Just to lay the context, I feel like this new generation of cancer research really started with checkpoint therapy. And one event which caught a lot of people's imagination was, Jimmy Carter. He was like 90 plus years old. He had melanoma. I forget the year, but what happened was he got the checkpoint therapy and he recovered from melanoma. So that was almost a watershed moment, both kind of scientifically as well as in the culture, I feel, where, okay, so there's this new kind of wave of drugs which are possible and kind of the cure for cancer, we may be getting, very, very close to that. So that's checkpoints. I want to talk to you about 2 classes of drugs and how they work, but I won't go into any more detail than that. This first class of drugs is called antibody drug conjugate. And the way you can kind of simplify that in your head is think of it as heat-seeking chemotherapy. So chemotherapies, most of you know how they work. You pop a pill, it goes after proliferating cells in your body, and it kills those proliferating cells. But, that's not ideal because it's indiscriminate. It will make your hair fall. It will make your, you'll just be very sick because of these things. But what if you have an antibody, which is just this classic protein structure, and on the one end of it, you have, the antibody seeks the antigen which is specific to the tumor. So, for instance, in the lung, there is a antigen expressed in NSCLC tissue called nectin. So if your antibody is designed to go connect with nectin, it's just going to find a way to go reach that nectin. But along with that antibody, you have the payload. And this payload is basically your chemotherapy. So instead of like dosing the patient large amounts, systemically, this is a way for you to reach the drug in high concentrations into the site of action. So, this is still kind of an exciting area of development. And recently, last year's ESMO was really, there were some very exciting results. So this was in bladder cancer, if I remember right. And what you see here is classic curves you see in clinical oncology. So this is the start size of the tumor. They normalize it so that everybody's tumor starts at 0. So anybody whose tumor shrinks, they have responded to the therapy. Anybody whose tumor grows have not responded to the therapy. So remarkably, in bladder cancer, they saw complete response. Complete response means your tumor shrinks by more than 90%. It's just not something that they've seen before. So this ADC was really quite miraculous. Yeah? Yeah. Yep. Exactly. So both of those are very good questions. The first question, I'll argue away and say that, in the beginning we talked about 3 buckets. When you're in the middle of drug development, you're rarely thinking about whether this drug is kind of economically the most viable thing or not. So that's one thing. But on the other hand, improvement depends on that, right? Chemotherapy today, people take all kinds of toxic cocktails, people have all kinds of negative effects, people sometimes have to stop their cancer therapy because they have intolerable side effects. So this is an improvement worth making. What is the economic cost of that improvement? I'm not, that's like a very different question that I'm unable to answer, but the improvement is worth making. The second question you asked is a very important one, and there's all kinds of design issues in ADC, and that's the excitement for drug developers, because we have to come up with newer and good designs. And obviously the ideal design is an antigen which is 0 expressed in normal tissue and 100 expressed in tumor. That doesn't exist. So how do you kind of make the affinity stronger? We can think of the antibody being connected to 2 antigens in the tumor so that you limit the risk of exposure to normal tissue. So there's all kinds of innovation that you can come up with there, and protein engineering is just like miraculous these days. So there's all kinds of interesting things you can come up with. In particular, you can decide what the target is, you can decide what the payload is. In fact, chemotherapies which have been rejected in the past as being too toxic to be given systemically can be given on ADCs because you're kind of focusing it on a particular area and a particular cancer. How deconjugatable should that linker be? So there are all kinds of interesting questions that you can ask. How many payloads? So that's something called drug antibody ratio. So how many of these payload molecules do you want to do you want this antibody to ferry to that location? So all kinds of interesting questions remain. The Enhertu, I think, was a drug that had this miraculous effect last year. A lot of investment and energy is going into the development of ADCs in pharma today. Next, this is even more kind of futuristic and mind-bending. So this is T-cell engagers. So as you all may know, the kind of next frontier in attacking cancer is channelizing your own immune system, your own T cells, your own cytotoxic cells to go after cancer antigens so that they can get rid of the antigens. Checkpoint therapy itself works like that. But what this therapy T cell engagers do is like this. Again, there is an antibody. Again, very much like the ADC, one side of the antibody will attach itself to the tumor, and on the other side, instead of having a payload, it will attach to a T cell which it can activate. So basically what you're trying to do is you're trying to drag all the warriors in your body to the battle site where the tumor is, and then you're going to take all of this and dump it there. And again, there's been some good success over the past years there. There are some concerns about this drug as well, and this is something that anybody that has modeled the immune system will have confronted at some point, and this is true even for infections. If your immune system activity is too low, then you'll just die of the infection. You're just not responding well enough. On the other hand, if you over-respond, that's a problem, because that is what is often called as septic shock in the case of infection, or cytokine storms. So basically your immune system has not evolved to kind of like find that right spot all the time. In fact, most of the time when people died of COVID, they didn't die because of this, they died because of that. Because your organs shutting down and you go into the ICU and die. So there's always this sweet spot that you want to trigger this to. So the biggest challenge in kind of developing T-cell engagers is you want to activate the system enough that you get good at killing the tumor, but you don't release too many cytokines. So there's all kinds of interesting things. My next case study was going to be that, talking about how you can kind of, what is the dose you want to give so that you see good efficacy, but not that much safety events. So really you want to like hammer the immune system in the sweet spot and not go too high or too low. I'll just talk to you a little bit about what that case study was. So basically a small biotech came to us and they had preclinical data. So they had data from monkeys, which means they gave this drug at higher and higher dose to monkeys, and they had a dose-dependent increase in the cytokine storm. They had efficacy data from a mouse. So this mouse was loaded up with a xenograft of a tumor, and then they saw what is the dose that they needed to get rid of the tumor. So when you have these two different species and you have this complicated dose line that you have to walk, you have to do a lot of clever things to bring the model together and make sure you kind of, translate appropriately from these species. So we were able to kind of help them with, what would be a good dose, and we gave them some predictions. And, dose is not a single thing. We recommended that they kick the immune system, and this is not, the most T-cell engagers follow this kind of stuff, which is, kick the immune system a little bit, get a little bit of IL-6, get it out of the way, so that in subsequent hits, you're done with all the toxicity, and most of the efficacy is, goes for tumor killing. So anyway, it's a complicated case study, and I'm not going to go through that right now. Yeah, so this is the group, and I'll come to that in a second. And I'll pause here so that if anybody wants to look at some of the publications which talk about how modeling can support T-cell engager development, that's right here. Okay, so I'll move on to this case study, which I think is kind of, like I said, high impact, and where mechanistic thinking motivated us to look at clinical data in a different way. So before I go too far, any questions on ADC or T-cell engager? I know I ran through it pretty fast, but it's exciting, and so the next time you look at a news article for either of those, I hope you're intrigued. Any questions on ADCs or T-cell engagers? Okay. So let's change gears and talk about what happens in checkpoint therapies. Have any of you heard of checkpoint therapies? You don't count. Yeah. Pembrolizumab, ipilimumab, nivolumab. Okay, so I'll talk to you a little bit about what exactly is happening here. So this is a very famous paper called on the tumor immune cycle. So basically, if you have an understanding of the immune system from infectious diseases, the immune system has evolved to understand foreign material. So it's the same sort of programming that it uses to understand what happens with tumors. So basically, when a cancer cell releases antigen, that is foreign antigen, and in principle, that can tickle the immune system to go through the process of, innate immunity, releasing T cells, and those T cells can be, come back to the tumor microenvironment to really get rid of the tumor. So this happens, and if you read the history of cancer, there's all kinds of shocking things that people did before the era of chemotherapy. So in the old days when people had melanoma, they used to kind of scratch that up and put all kinds of bad like infectious agents in the melanoma tissue to activate the immune system. And I read this in the book The King of All Maladies by Siddhartha Mukherjee. Yeah. Emperor of All Maladies. Yeah. So basically, the fact that the immune system can go after cancer is not unknown. That was very well known. But in the era of chemotherapy destroys everything outside. So it used to destroy, even the kind of friendly fire coming in from the useful fire coming in from the T cells. Even all of that gets destroyed when you go into straight up cytotoxic therapies. And the tumor is not sitting still either. The tumor is very clever. Most tumors evolve into strategies to slow down the immune system. So that's called a checkpoint, and I'll talk about that in a second. So basically, the immune system has evolved so that every time it gets a rev up signal, there's always a possibility to get a break signal as well. And this also evolutionarily is a highly redundant, highly pleiotropic sort of system. There are many signals which rev up the immune system. There are equally many signals which slow down the immune system. So one of those slow down signals is PD-L1 and CTLA-4. And basically what the tumor does very cleverly is it will start presenting these break signals so that any T cell, even if it's revved up and comes to the tumor site, will see all these break signals and quiet down, and the T cell will become useless in the tumor microenvironment. So what the checkpoints basically do is break that interaction. So that's why they call checkpoint inhibitors. This is a checkpoint. It slows down the T cells. A checkpoint inhibitor slows down that interaction. So the T cell which is activated remains activated in the tumor cell and can potentially lead to tumor cell killing, and that's basically the theory of how checkpoints work. Okay, so this is what's happening in the cell to cell level. Are any of you kind of familiar or read cancer clinical trials or look at clinical trial data from cancers? So that's a whole other scale, right? So this is happening in that small scale. Again, y'all are all living inside the cell. What I'm talking about is two cells interacting. But there's a lot of, when you look at a clinical trial, how do you interpret what happens there? So usually when you look at a cancer clinical trial, you look at progression-free survival. So this is a typical Kaplan-Meier curve. Any of you would have read, seen it in any epidemiology or stats course. Basically, when you have 100 people with cancer, as they keep falling off the trial, that means they failed on the trial. At the end of the trial, only 50% of the people have been benefited by the drug. So that's progression. That's disease progression. And when this is the progression-free survival curve. So this is the concept of disease progression. Basically, you're trying to say that is my drug benefiting the patient or not, or are they progressing on cancer? Now, clinically, what happens when a patient comes into a trial or into the clinic to get therapy for cancer? It's not a single average lesion. A person with melanoma can come with multiple lesions. These lesions can be in different parts of your body because usually people come in with metastatic melanoma. So you can come in with melanoma and you can have a lesion in your lymph node, you can have a lesion in your liver. So when people come in, they have multiple lesions. So when a doctor tries to treat this patient, what they do is they track some of these lesions. Patients can have 10 lesions, 15 lesions. So what they'll do is they'll target 2 to 5 of those lesions and call them target lesions. And in the context of a trial, they'll say, when I give the drug, how much do the diameters of my target lesions shrink? So as you can imagine, you kind of flatten that tumor out into like a sphere, and then you say, if that target lesion shrinks, then I'm doing well. But the important thing that we do is we sum all of the longest diameters of the target lesion and say, does that shrink? There is one more, and if it doesn't shrink, you're progressing. If it shrinks, you're lucky. But there's one more concept in cancer as well, which I think many of you kind of worry about from that cellular angle, which is metastasis. So people come into the clinic, they have 10 lesions, the doctor will spot 5 lesions and he'll say, I'll see how it shrinks. All 5 lesions may shrink, but you may get a new metastatic lesion. That means you're still progressing on therapy. So there are many paths to progression. Your target lesions not responding, you're getting new lesions, or your non-target lesions are growing. These are other reasons why you could get progression and fall off that nice curve that we want you to be on. Okay. Now let's go back to physiological concepts, right? So people progress. Why would some lesions respond? Why would some patients respond? Why would other patients not respond? So among cancer lesions, I work more in melanoma, so I'll talk about melanoma. So there's a concept of cold lesions and hot lesions. So hot lesions are lesions which are very visible to the immune system and the T cells are flooding it. So you already know that, when you have a cut or when you bruise yourself, sometimes that area becomes red and warm. So that's basically because all of your immune cells are active there. So you can kind of think of it as in an immunologically active site. Whereas a cold tumor is something that has hidden from the immune system and there is just no immunology there, so it's a cold lesion. So already you can imagine if you put a checkpoint inhibitor here, there is some usefulness. You're taking the brakes out of the T cells. These T cells can kill these hot tumors. But if you put a checkpoint inhibitor there, it's fairly useless because there is no T cell whose brake you're removing. So there's already, you can start seeing that there's some heterogeneity in those lesions which will affect the way this drug works. And clinically, how does that translate? It translates like, you can sometimes think of patients, all of their lesions are cold, all of their lesions are hot, but as you can imagine, real world is never that simple. Most patients present like this. Some of their lesions are hot, some of their lesions are cold, and so there is heterogeneity in the response to the therapy. Now I'll go back to looking at the clinical trial, right? Like I said, if you're lucky, this is the response to, I think, Pembro. If you're lucky, your tumor, which, everything is normalized to 0. If you're lucky, your lesion drops off to 0. Your sum of longest diameters, it drops off to 0. If you're unlucky, you're growing here. So this is how people usually look at trials. And they say, oh, these people are all responders and those people are all non-responders. But what we believe is that kind of shoves under the rug a lot of important heterogeneity. If you look at the sum of longest diameters, you may see that, it's kind of steady like this. But what we believed was happening based on theoretical considerations was that, the sum of longest diameters, the average behavior looks like that, but that could also mean some lesions are growing and some lesions are shrinking. In particular, cold lesions may be growing and hot lesions may be shrinking. So is this merely a theoretical consideration? Is there any evidence for this? So this was work that we did with Merck. So actually I should explain what a waterfall curve is before I go to that. So, okay, so there I showed you how, the tumor diameters look. This is the tumor diameter at the end of the trial. So if you look at the tumor diameter at the end there, and you stack all the patients, these are the patients that didn't respond, and these are the patients that did respond. So this is how the patients look. And if you think of this, you can be like, this guy is very lucky, that guy is very unlucky. So this is what, you stack those results in what is commonly known as a waterfall plot. But what we observed when we looked at data from Pembro was the following. So when you look at that waterfall, this patient who was a very good responder and on average their tumors really shrunk, they had individual lesions which were growing. So these guys sooner or later would have dropped off the trial because this waterfall is caught at a particular point. So sooner or later these tumors would have grown and these tumors would have affected the patient. All the way on the left hand side, you see patients, you're like, oh, these patients, they're really not responding to Pembro. But even in these patients, there are many lesions that respond. So based on looking at the average behavior, if you say, oh, this guy is not responding to Pembro, I'm going to pull him off Pembro and put something else, you run the risk of losing the response that you're seeing in some of these heterogeneous lesions. So, there is heterogeneity in lesions, and that's really important. And why is that important? Like I said, that's important because it's important in patient care. So for a patient for whom every lesion is growing, it's not a big mystery. So, you need to take this patient off Pembro because they're just not responding. For a patient whose every lesion shrinks, that's also not a mystery. This patient is a good responder to Pembro, there's not a problem. But when you have a patient who's somewhere in between, you really need to be careful about how you're making that decision. And this decision is usually made by something called RECIST score. I'm not going to go into a lot of detail there, but suffice to say that acknowledge patient heterogeneity in lesions before figuring out what therapies they have to be on. And before I go too far, so we're not the only group that came up with these recommendations. There were several other groups that came up with these recommendations just looking at clinical data. But the point that I wanted to make was our group was a modeling group. And the reason we kind of, came up with this hypothesis and had a chance to test it on data was that we developed these models. And we already said, this is one of those QSP style models for what happens in a tumor microenvironment. There's tumor, there is T cells which kill the tumor. But the minute we made such a model, we knew that we had to parameterize it using hot and cold lesions. And we already knew that trying to fit the data, we had to factor in heterogeneity, otherwise we're not able to fit that data. So once our model tried to fit that data, and we proposed that there are heterogeneous lesions and we have to factor that in, we came up with a proposal for the field to, when they develop these models to study clinical trials, what should those models look like? Usually those models you can think of as a single prototypical lesion. So most cancer models, even when they're trying to kind of support drug development, most of them will be the drug went into the tumor, this is a single average tumor, this is our prediction for what will happen when you run a clinical trial. But our claim that we made in this paper was, you have to look beyond a single average tumor. A model should have a single prototypical lesion, but you need to have, when you come up with that structure of a virtual patient, you need to account for these multiple lesions. You need to account for other paths to progression, which also include metastases. And that's how you come up with a virtual population that you can test your new drug on. If you flatten that complexity with a single lesion, you're missing out a lot and you're likely to come up with inaccurate advice because you're not capturing the heterogeneity in the lesions. So, I'm not going to go into more detail here as well, but this work was done by Kannan, Madhav, Mritika, Aparna, and several other colleagues at Vantage, and Brian Top and other colleagues from MSD, and I'll show you the publications where we can see all of the colleagues. This is also a picture of Vantage that I should have probably showed before. So this is our team at Vantage. There's about 40 of us. This is, we've been in business for about 10 years now. These are some of our clients that we have published with. We've worked on diabetes, autoimmune disease, cancer, etc. So, and a handful of colleagues that we haven't published with as well. So, not showing all the names. And this work, where we're making the claim that lesion-to-lesion heterogeneity affects interpreting clinical outcomes, we have, a decent list of publications, all the way from modeling studies to clinical studies to opinion pieces that I'd, if you're interested, you should definitely look at. Okay, so that's a little faster than I planned, but I'll pause. Any questions? Did that sort of make sense? I think I blew past too much of the detail very quickly, but any thoughts? Even in your own studies with like cells and such, I think, most of the times mathematical modelers tend to average a lot of behavior, and that's usually very, very useful and effective. But the point is biology is complicated and you can't always average stuff out, and that's what we learned from, this work. Okay, that went faster. Yeah, sorry. Yeah, that is exhaustion. So now you're talking about T-cell engagers, so I'll go back to that. That's exactly right, that is exhaustion. So again, for those of you that are interested in, various, stages of cell maturation and cell phenotype, this is the question that you're asking, right? So this is the T-cell engager. On one side, it connects to a tumor cell. On another side, it activates a T-cell. The thesis is that the T-cell can exist in various phenotypes. When it's highly activated, it can be very good at cytotoxicity, but it can also release too much cytokines. If you kind of push it from an activated phenotype to a slightly exhausted phenotype, you don't want to push it all the way to a completely exhausted phenotype, but if you push it to a slightly exhausted phenotype, it can, so the first time you activate it, it's very ready to release a lot of cytokines. But if you keep knocking at the T cell, it's still pretty effective at killing, but it'll start secreting less and less cytokines. So the dosing game in T-cell engagers is to give non-toxic levels of hit so that you get rid of the cytokine response in the beginning, and then what you're left with is a way to reach the efficacious dose. Still effective state. That is the CRS, the IL-6 is the clear marker for that. So, and the way these trials are conducted is you first start with the slowest dose and you do a dose escalation. So you don't directly go to the highest kind of most dangerous territory. You start with the lowest dose and then keep going till you're hitting that, tolerable levels of CRS and reasonable cytotoxicity. So it's a challenging game. Where modeling helps is, so if the tolerable dose level is, say 30 micrograms. Without modeling, you'll start at 1 and then you'll double to 2, 4. So you'll spend a lot of time trying to get to 30. So what modeling helps is to say that, hey, below 10, you're under really no risk. So that's the sort of stuff that we help with. But eventually, at the end of the day, it's the clinical trial and, you have to learn a lot from that trial. Yeah. So the thing with T-cell therapies, including CAR-T, is officially these guys could get memory of that antigen. So it's possible that once you treat them like this, it's not like chemo, you sort of like treated them for good, but remains to be seen. We've not had enough experience with T-cell engagers to say that, it's one and done, or whatever you shrink it and done. It's possible. I don't know enough about that literature, about the etiology of metastasis and whether the immune system plays a role there. There's all kinds of selection pressure, right? So if an immune system gets rid of the susceptible cells, then what remains will grow. So I understand that literature a little bit, but really what types of biomarkers are kind of indicate higher probability of metastasis, I don't know the answer. Any other questions? It doesn't have to be about this session. Any other questions also I'm open to. Yeah. And surgery and radiation are standard workhorses. Like even with checkpoint, I think surgery and radiation will combine very well. But again, in the pharma development context, I don't know how well the photon therapy. I've heard, those radiation therapies work really well, but I don't know. Yeah, targeted radiation. Yeah, yeah. But again, I don't know enough about what's the cutting edge in those therapies. But I do know that again, the clinical trial in a pharma company is a very sterile environment, in the sense when you're trying checkpoint, you almost only try checkpoint. But oftentimes when patients fail on this, they go to radiation surgery and things like that. Or sometimes people come after they've gotten some of their lesions removed by radiation and surgery, and then come into for the non-responsive lesions. What kind of therapies? I guess what you're saying is, if it's an immunologically inactive cancer, if it does not present MHC, if it's a cold tumor, what would you do? So, of course, there's surgery and radiation and those kind of physical interventions. There are kind of new therapies which aim to turn cold tumors into hot tumors. So there are therapies which will kind of invoke the innate immune system, which is less specific. So there are things called sting agonists, for instance. So they may go into the lesion and release enough antigen that NK cells and mediators of the innate immune system come, and that may then trigger adaptive immune system to come. But I don't think, that turning hot to cold, it's still a long way away. Yeah. And of course, there's vaccines and things like that. So if you can, kind of really make it immunologically active in a way. But again, I feel like I've not worked on any of those yet. That's almost by definition. So anything which is immunologically active, what they call as tumor infiltrating lymphocytes. So you take a biopsy, and in the IHC, if you see the TILs, which are less than 1%, then that's classified as a cold tumor. Anything which is more than 5% or something like that, at least in melanoma, that's classified as a hot lesion, and anything in between is warm. Yeah. Oh yeah, there's a lot of work in health economics and outcomes research. So that I actually encourage scientists to seriously consider that field, because now it's kind of saturated with economists and policy type people, but it's a great place for scientists to work in as well. So there, the crux of the question is, how much should what is the exact benefit of a drug to society, and how do we measure it? And they usually measure it by quality life years. And so that requires an understanding of, if I give this RA drug, how is that making that person's life a little better? And you have to understand the science a little bit and how it adds to life. And then in a population, if it adds 100 years of quality added life years, how much should I price that drug at? So that's kind of like a super flattening simplification of that field, but there's all kinds of interesting modeling, which also considers how the drug acts in determining how to price that drug. Yeah, so that's often called a phase 4 trial. That is, your drug has been approved, but because of regulation or other reasons, you're monitoring what happens to that patient. So that's post-market surveillance. Like I said, I feel like I've not looked at enough of that data, and I would have loved to look at more of those data. And the reason often is that data is not as clean. Clinical trial data itself can be very messy, but real life data is much messier. So oftentimes, what do you want to take from that? People don't usually go on fishing expeditions in that data, but I do feel there's a lot of good work that can be done and extracted from that data. Then also you will drop off the progression-free survival curve. So people drop off not only because the drug is not efficacious, but they may drop off the trial if they have intolerable side effects or something else happens. So patients do drop off, and that is counted as progression if they drop off because of side effects. Checkpoints, we've been lucky, especially with anti-PD-1, anti-PD-L1, that the side effects have been very tolerable, and the condition, of course, you want to get rid of your cancer, that I think the people who drop out because of unspecified reasons or talks is not as high as some other trials. Yeah, so a famous case is this drug called Vioxx, which I think Merck also made like somewhere in the aughts. So this was a drug which was a pain medication basically. And in the clinical trials, there was not enough of a signal. But the clinical trials were like, nominally clean and there was not much of a signal. But when the drug was administered to the, large population, we started seeing cardiovascular signals. So a signal that you don't see in the 100 people that you do in phase 3, you may see that signal in the real world when you dose to 1 million people. So drugs at that point, they are regulatorily mandated to be monitored. And when you see idiosyncratic effects or 1 in 1,000, 1 in million effects like that, you have to investigate. And if it's a very bad effect, you have to withdraw it. Sure, sure. Yes, yes. So the interesting question is what should be a combination? So that paper that I had, and maybe some of you will go look at it. So a few years back, combination of checkpoints was considered kind of the next big thing. And the claim that we made in our paper was, if you have a cold lesion, if it's not responding to your first checkpoint, it's really not going to respond to your second checkpoint. So ideally, you should have some orthogonal therapy to go after those lesions. And yeah, so exactly. So usually when patients don't respond, they take into something else. Usually they combine with chemo or something else. But yes, combination therapy is standard in cancer. Yeah. Yeah. So that is exactly what we did here. So we developed this model and we calibrated it to mouse. And when you transfer from mouse to human, first of all, you have to scale those compartments to kind of human-sized compartments. And there are physiological differences between mouse and human that you have to factor in explicitly. Mm-hmm. So you have a like supervised learning you're doing, and the output will be. Okay, so maybe I'm not sure I understood everything that you said, but this is this is basically what we did, right? There is one model. So there is PK, there is physiology, there is tumor, there is one effect diagram, and that we believe is common between mouse, monkey, and man. So identical mechanisms happen in mouse, monkey, and man. But we parameterize it differently for mouse. So for mouse, that plasma compartment where the drug goes in, that will only be a few ml. The receptor density in the mouse, we get it from what is there in the mouse lesion, yada, yada, yada. So we have a bunch of, the model parameterization is specific to mouse. But what we learn from the mouse is the cytotoxicity of that single trimer. So okay, so how does that drug work, right? There is T cell plus antibody plus tumor antigen. When this happens, the tumor antigen, you get rid of the tumor antigen, and maybe there's some recycling of the T cell, but we won't worry about that for a second. So what we get from the mouse is the dose response of this trimer and the kill rate. So we get that dose response from calibrating to the mouse experiments, and then we use that dose response to predict what is the efficacious dose in human. So that's basically what we do. And similarly for monkey also, it's trimer versus IL-6. So then we get that, a single trimer we assume behaves similarly in a mouse, monkey, and human, and what is the dose response of that, as you form trimers. But the rate at which you form trimers, the number of T cells, all of that could be different among the species, but once you have that entity, that behaves similarly across species. So that's the assumption with which we work. Okay, so if we have time, maybe we'll just like goof around with that video game that I came up with. So please set your expectations modestly. It's like not very fancy. Mohit had said this is a workshop, and I felt bad coming with just a bunch of slides. So I asked my colleagues to come up, show what they had. So let me see if this works here. So just to set some context, our colleagues at Merck, they worked with literally a video game company to come up with this toy thing. And their goal was, if we wanted to talk to other people at Merck, if we wanted to talk to clinicians about this heterogeneity thing, how would we share that information? So, okay, so like I said, this is like a little bit of a simple tool to show what all could be different among lesions. So I don't know who has it on, but if you look at, maybe I'll start again. Okay. So they're basically multiple modules, right? So, so here's the other thing, right? I kind of focused on heterogeneity being really driven by the difference in the immune contexture, but there's all kinds of heterogeneity. And I also assumed that a single tumor is a monolith, and that's also not true. There's all kinds of variability even within a single tumor. But, all that said, these are kind of other ways in which tumors can be different and can lead to difference in response. So, you can add a tumor. So this is basically like thinking of, what does an average melanoma lesion look like? And, it's nice to imagine that, it's small. It's the size of a marble, and some lesions can be very small. 5 millimeter is usually the limit of detection, and some lesions can be very big. And let's see. Okay, we don't have to go through all of it, but in this module, you can really play with the various tumor sizes. And then if you go back to the next module, oops, and I feel like I don't know how to handle this well enough, but if you go to the next module, it'll sort of allow you to change the tumor growth rate, and that's pretty important as well. So a therapy which works well in a slow growing tumor need not work that well in a fast growing tumor. So if you look at level 3, that's where, the whole cytotoxic lymphocytes that I was talking about, that starts to make an appearance. Some tumors are infiltrated with cytotoxic lymphocytes, and when you activate these CTLs, it allows them to kill the tumor cells, and activate, as I said, is really removing the checkpoint, the checkpoint that the tumor is expressing. So here you can make it a hot tumor, and so if it's a cold tumor, there's literally zero tumor cells, so that's 0%. So it's warm, and you can continue with that, and you can make this lesion hot, and this lesion is also hot. At one point, it used to become a red thing, but I guess it doesn't now. So there you go. So you plop in some Keytruda on it, and you can kind of see what happens. So that's the Keytruda. Yeah, there you go. It goes a little hot, and you simulate the trial. So behind this is a model very similar to the one that I showed in that publication, and you can look at it to see what really happens. And this allows you to simulate that trial for 6 months and see that the hot lesion is really shrinking, and the cold lesion is relatively unaffected by Keytruda. So the overall response of the tumor is determined by the CTL density. So this is the crux of the argument that we were making, and this could be the status of a single patient, and how different they could be. So I don't have to like walk this through and sit here and do it. So you guys are welcome to play with this, and hopefully delete it by tomorrow morning. Like I said, I don't own the software. The people who made it were nice enough to share it with me, and I told them I'm going to share it with a bunch of scholars. So do play around with it. But if you take away something from this talk, what I'd really like you to take away is when you look at a cancer trial and when you see that trajectory, that is not, kind of the overall response of the patient. There's a lot of heterogeneity going on. There may be some lesions which respond and some lesions that don't. Okay, with that, I will pause. I don't know enough about the relationship between telomeres and cancer. I know there is, that is, the older the cells are, the more likely the, every, proliferation leads to more mutations and things like that. But really what you're thinking of is disease etiology, right? Like where does the mutation come from? And I don't know enough about that. Thank you for the opportunity. Yeah.